Many years ago, I went to the San Diego conference. I can't remember what year it was, the one before the most recent one, and I was really inspired by all the lecturers there. So I'm glad to be in cahoots with you here. And I just want to shout out to Jim Mouseth, who inspired me back then in my lectures. So thanks a lot, Jim. And know that you've affected a lot of students too. So. Yeah, so I'm here to talk about propagation, and basically the big picture here is I want to encourage more of you guys to try propagating succulents, one way or the other. Okay, there's a lot of really cool plants, and I think by trying to propagate them and grow them, you get a lot more feel for the plants that way, and it's just really fun to experiment. Okay. So I really recommend if you want to get more into propagation, get a copy of this book here, Plant Propagation by Hartman and Kester. And what's really great is that you guys aren't undergraduates. You don't have to buy the most recent copy, so you can buy the more affordable 6th and 7th editions. Okay. Anyways, really good book. When I started teaching students, I never took a class on propagation. I took botany classes. I picked up this book and I read a few of the chapters so I can teach students sort of, basically get more information to teach more to other people. So highly recommend that copy of that book there. So I'm going to reference our soil that we use in growing plants and propagating some succulents. And I said some because we don't use these for all of them. But anyways, I want you to see what it looks like. Here's this is our succulent mix right here. I'll give you the components of that here in a little bit. Another component that we use sometimes is horticultural grade sand, 12 mesh. That's really fairly coarse sand. Okay, it's in our soil. Should it be in our soil? I wonder about that. But I like the weight it gives some of our containers because succulents can be top heavy. Okay, and then, so there's our, this will get here in a sec. But this is what I use for some plants, and when I talk about aloe propagation, I'm going to bring this up here. 1/8 inch sifted red lava, okay? Our soil mix has, our UCLA soil mix has 3 parts 5/16 red lava, and then what I take when I buy this red lava is I sieve it out, and I get this for propagating some succulents like aloes. What you can see is that our soil mix is very low in organic matter, so that when it breaks down, it's basically, when the organic matter breaks down, there's even more inorganic materials there, and so you get a very airy, porous container mix that's really good for succulents, right? Okay, succulents like oxygen, they don't like too much water. I forgot to bring out my sticker that Sandy here gave me last night that says rot happens. Okay? All right? And you want to avoid rot. So anyways, especially when you're propagating. We'll get back to that here in a little bit. So yeah. All right. So just a little quick thing about growing stuff from seed. And this is really general. We can have discussions afterwards. Anybody out there that disagrees with me this? But it's just really general about growing stuff from seed. Is that Mediterranean climate plants, you typically want to plant them in the fall. So, and this is what I'm thinking, like South African bulbs, Othana, some of the South African summer deciduous succulents, and then some of the succulents from the Sonoran, northern Sonoran, they're best planted under these sort of temperature regimes here. Okay, and we'll talk about water here a little bit more. When there are succulents that are sort of ones from the more tropical areas, and I say tropical, which in quotes because subtropical plants also included, is these plants, when you germinate their seeds, you want them a little bit warmer in the 70 to 80 degree temperature range. And so like this is aloes, we'll talk more about them in a bit, Dorstenias, Welwitschia, I'm going to show you guys Welwitschia propagation, talk about stapeliads, and then other cacti. So, Milo cactus that Marlon talked about yesterday is from Brazil, somewhat tropical. So that one's going to be different than if you're growing, let's say, a barrel cactus, a echinocactus, or even, anyone ever here tried propagating pedicactus from seed? All right, a couple people, great. I'd love to talk to you guys about your successes and challenges with that later, okay? And then there are plants that are from summer rainfall areas. where they really do their stuff in summertime. And so like Lithops and Ariocarpus, whenever I plant those seeds, I do them in early spring, okay? And for us in Northern California, early spring is something like April, okay? April, May is when we'll plant those seeds. So they get it. So their first year, they get an extended summer of growth, okay? So whenever you're germinating stuff from seed, you want to make sure that your pots are not in direct sun oftentimes, because you can bake the soil and dry things, and constant moisture is really important for good germination. I'll talk more about that. So in places where it's warm, and even in my greenhouse where it's warm and sunny, I'll put my seedlings somewhere where it's a little bit shadier and there's not so much direct light beaming on the pots and drying things out on the surface. Okay? By the way, my style is questions as I go along. So if you guys have a few questions, I'm going to pace myself, etc. But I'll take a few questions as we go along if necessary. Okay? Here's some Othanas. These ones were done last winter. There's the date 11/23/16. These ones I planted them on the surface of the soil in the winter time, and I just kept watering it till they germinated. And then once they germinate, then I cut back on the water a little bit. But these are now are dormant for the summertime. Okay. So Othana Hooray on the right, that really beautiful Othana there. Okay. So again, these are winter growers. So I plant these seeds in the fall. And for us in Northern California, I have to do it sometime about October or November. If I do it too early, we still have the heat in September to deal with. So, yeah. Okay, this is not the conservatory at UC Davis. Okay, this is the Gardens by the Bay in Singapore. I guess it's part of a big, casino, etc. there. Lots of money, the gigantic greenhouses. Anyone want to help me build a new conservatory at UC Davis? I'd love to talk to you about that. Okay? Ours only have 14-foot ceilings, and so this Dorstenia that's looks like it's about 8 foot tall in here. I envy the size of that plant. Okay? And if you guys haven't been to Petra Christ's rare succulents in Rainbow, she has one that's approaching this size, a beautiful specimen of it. They came from us. Okay? So I'm going to talk about propagating this plant here. So here's one of our larger specimens. It's about 4 feet tall in a container. Needs a bigger pot, obviously. Okay? But a lot of people always ask me, when will my Dorstenia flower and when can I pollinate it to make seed? Well, good luck, because I've grown a lot of them and I have only about 3 plants that have sex with each other. Okay? And that's about as non-PG as I'll get, okay? All right? So, so it turns out that the plants, sometimes the plants act like females, sometimes they act like males, sometimes I don't even know if the pollen works on them. Okay? But here's stages of the blooms, the inflorescences. This is not a single flower, this is many flowers acting like one. Okay? And this right here is the female stage. All right, and this is when you can pollinate it. Look at the pollination stage. So in my plants, when I'm trying to pollinate, this is the stage that I go after to pollinate them. You can see the stigmas sticking out right there. Okay, and then as the flowers transition, they start making the male parts, the stamens, in there. Is it still fertile? I've tried pollinating these from here to here, and I haven't done enough controlled experiments to say this is when it works, this is when it doesn't. Okay. Very challenging when you have limited plants to work with. And then you get to this stage right here, and this is where the plants are dumping out their pollen, and by this time they're basically, these are male flowers, and the females are no longer receptive. You can't pollinate them anymore. Okay? So, it's kind of fun, huh? All right? So here's that plant again. Okay, and in this situation here, go back for a second. Oh, I guess it transitions in. What I was trying to do is I was trying to keep track of who Daddy was when I was pollinating these. Okay, so that was, dad number 1, dad number 4. Over here's number 2. Okay, and what I found is that there's some plants that are very female. They're reproductive, like this one right here. This one is, I think this was what they call a UCLA clone. And this one can make seed. But there's other ones that, that for some reason the pollen does and doesn't land on this and germinate. Anyways, germinate, fertilize the ovules in there. And so honestly, I haven't figured it out. I'd love to try to figure out which plants, what's going on with the plants, but it's kind of a plug and pray type thing, okay? Or pollinate and pray, okay? Yeah. So here's when the plants are going reproductive, okay? When, we pollinated, and now it's making babies, the seeds in here. And these, the way I describe these are somewhere between a missile launcher and really good whiteheads to squeeze out, okay? Some people like that thing, right? Anyway, so they squeeze them out. It's actually like the seeds develop between what are probably the sepals inside of there, and they squeeze it like you do a watermelon seed. They squeeze the seeds out, and so I use this tule fabric here. This is very good stuff for a horticulturalist, okay? To capture the seeds in there. I make these little mesh baskets and then peel them off and dry the seeds a little bit and then plant them up. Okay? In fact, this is not Dorstenia gigas, this is Dorstenia bornemiana. Here's the seed planted. Okay, and what I do with these and some other succulents is I plant them in the pot, with succulent mix all the way up to the top, put the seeds down, and then put the layer of sand on top. Okay, water them in, and for nearly all succulents, in fact, nearly all plants, constant moisture until they germinate is really important. Okay, and how many of you guys have potted some succulents up, and you go there every day, some seeds, and you go and water them every day, right? And then you're like, oh, I forgot today. No, the soil dried out. Well, I call this the Ziploc baggie technique. Okay. 3-inch pot in a quart-sized Ziploc, water it in, let it drain out, put it in the bag. You see how sweaty it is in there? No watering required. Constant moisture. And all you got to do is you got to watch out until they germinate, and I'll talk more about that here in a little bit. Okay? I encourage you guys to try propagation. Okay? Again. Here's them germinated.
Ernesto, do you seal the bag?
Yes. Yeah. You seal the bag up. Yeah. Close up the bag, yes, very nice and tight. Sometimes just to make myself feel better, I give them a good shot of carbon dioxide and breathe in and say, germinate. All right. So there's a pot of seedlings right here. All right. And I'll talk more about this a little bit more, but I like to let them get nice and crowded and as big as possible before I separate them apart from their siblings. Okay. Until they get old enough that they start fighting, right? They don't fight when they're really early, it's a little bit later. Anyways, actually it varies, right? So I did one year, I had so much Dorstenia seed that I decided to try doing a little experiment, a visual experiment. 50 seed in the way I described before, seed on the surface, sand on top as a mulch, or inorganic mulch. Right there, and then here's 50 seed that I planted, same thing, without the bag, and I water this every day. It's obvious which one's better, right? Okay? I don't know if that's, research publishable material there, but to me it's a nice visual of constant moisture versus not. Okay?
Do you sterilize the soil?
Good question. Thank you for asking. Yes, we do sterilize the soil. We take our soil and we pasteurize it for about 2.5 hours in a big autoclave. But there are recipes for pasteurizing the soil in microwaves. Okay, or if you have an old pot that you don't need too much, moisten the soil, put it in the oven at low temperature. You basically want to keep the soil for about 30 minutes to an hour at about 165 Fahrenheit. Okay? So really low setting on the oven for about an hour or two to sterilize your soil. It's really important to have the soil be fairly clean of other things like mosses and algae, fungi, fungal spores. So I'll talk more about that a little bit later as well. Okay? Any other question about that?
Ernesto, if you heat your soil, then afterwards do you have to let ammonia diffuse out?
Good question. So you heat the soil and then you let it cool down and sit for a while before you then plant into it. So it's good to do that ahead of time, the day before, or a few hours ahead of time. We have our soil done in big batches, and then we store it nice and clean for when we use it. So yeah, sometimes the same, we'll use the soil months later to plant up, but we keep it relatively clean.
What kind of water do you use when you do your watering?
We use so a little bit of water with fertilizer in it. So basically whatever fertilizer you use, about a quarter strength of it. Okay? And for seedlings, they don't need a lot initially. Our soil has a little bit in it. So yeah, it's about a quarter strength. You know, the cleaner, the more pure the water is, the better. But you know, for seedlings, tap water with a little bit of nutrients should be fine. Okay?
Do you not worry that these seeds might need mycorrhizal fungi to germinate?
You know, that's a really good question. But my impression is with things that need mycorrhizae is that really it's when they get a little bit older is when that comes into play. And so the most important thing when you're first planting them is keeping fungi and bacteria from causing rot to happen. So so having clean soil to help seeds get along better is better than trying to put some mycorrhizae in there. And in fact, when you take seeds like from a cactus, you want to have it be clean to wash off all the sugars that might be on there, so that there's the least amount of food for fungi and bacteria to have a party at your expense. Okay? Yeah. So here's some of the seedlings of Dorstenia gigas in the pot there after they germinated. Still in the bag. You see droplets of moisture in there. Okay? These ones I can let them go for a decent while in the high humidity chamber and not lose too many of them. Okay? And these plants are leafy succulents. They like water, especially when they're young, and so constant moisture is really important for good growth. In fact, here's a couple of my most recent pots of seedlings right here. Okay? Ooh, right? Okay? And you see how big they are? These are already getting on, some of them are about an inch tall. And they were nice and crowded. And you know, when they're young, you want to keep them crowded as long as possible because most succulents are relatively slow growing compared to other plants. And if you pull them apart when they're too young, you damage roots, and then you really set back the growth rate of the plant. Okay? I'll show you a little experiment with aloes a little bit later that I did. There's my seedlings all potted up separately, you know, separated out. They look kind of consistently the same here. Okay? This particular batch, it seemed like Mama and Daddy were pretty good at making all the babies look the same. Okay? Yeah?
When you get to this point, do you ever have to deal with fungus gnats?
Ah, good question. Fungus gnats. Sorry, the question was, do I have to deal with fungus gnats? And it turned out that the sand, the mulch sand, is really good at deterring fungus gnats. For some reason, the sand, whether is it the color, is it that it's a little bit drier, the fungus gnats don't seem to be as active when that layer of sand is there. So one of the benefits for the horticultural sand as a little mulch on top of your seeds is decreasing fungus gnat activity. Yeah, it really does work. We have people that grow this thing called a Arabidopsis, a lab rat weed, and they use it. When we can convince them to use it, they get a lot less fungus gnat problems. And then you don't have to use fungicides, etc. Okay? By the way, here is a little diversity of the seedlings when they grow up, some of those same ones that you saw earlier. And look at the diversity of foliage here. Broad leaves, smaller leaves, bigger leaves over here. This one was actually variegated when it first came out, this Dorstenia, and it went kind of pale. You see how it's much paler than this one? It's not lacking nitrogen. It just turned out to have less chlorophyll. So that one's like, I love that plant. It's my special baby. Okay? Yeah. Just to show you, just two of the plants. These are some of the same plants in here. Larger leaves, smaller leaves. And this is the thing about when you grow stuff from seed, you get to see a little bit of the diversity of what makes a particular species of plant. Okay? And then when you hybridize, you get even more diverse. You get similar diversity or more. But these are species right here that are crossed with each other. So, yeah, that's the beauty of growing stuff from seed. And then there's just this. Okay, isn't that pretty? The little my predecessor at Davis, Tim Metcalf, called these the bunny ears of succulent seedlings. Okay, and this is a Caralluma acutangula. And then there's what's going to become succulent in here. Okay, for those of you that are into plants, this is the hypocotyl that swells up and becomes the big swollen part of the plant. Okay, look that up, hypocotyl. Okay, it's a really cool thing in plants. All right, and by the way, this is a relative of the Caralluma, of the Plumerias. Okay, and here, just to show you someone growing these in really fine mix, really good moisture, okay, with vermiculite, soil with vermiculite in it, and then a misting system to keep it nice and moist, all right? And then here's someone else using a humidity chamber. Some people like using this. See right here, this is really good fungus gnat habitat. So a little bit of layer of sand in there would really decrease the fungus gnat activity, okay? And then this is really cool, and I bet you there's some succulents you can propagate this way. Plumeria seeds in a little bit of styrofoam floating in the water. That's kind of cool, huh? Okay, and I bet you there's larger seeded succulents that you can do this to. We all remember the avocado with the toothpicks in the side? Okay, yeah, I bet you that, I wouldn't jam in there, but how about making a little like floaty for the seed, Yeah. Anyway, that's kind of cool. Okay, let's talk about a plant that sometimes that actually likes water and sometimes doesn't have enough to make babies, but because one of the previous speakers was talking about how plants always trying to reproduce, we're going to talk about Welwitschia and its the way it germinates. So here's a plant called the mother of all Welwitchias there in Namibia, near Swakopmund, that I got to see a few years ago. And for those of you that are young, you may not make a lot of money when you're growing plants, or working with plants, but every trip you take is a tax-deductible vacation. Okay? Yeah, Namibia. It wasn't free, but it was tax. I got a tax deduction, so there you go. Got this. This is back in 2008. Here's a relatively sizable plant. This one's a male in here. Look at this more a little bit. That's my rugged outdoor, explorer photograph. Okay. Thanks to camera angles, this one looks really huge. It's about as big as the previous one, but not as big as the mother of all Welwitchias. Okay. These ones where they grow in Namibia, all right? This is like a forest of them. Look, 1, 2, 3, right in there. This is, I jokingly call this a retirement community with no new members, okay? These plants, they try to reproduce. That's what plants do. That's what all organisms try to do. That's sort of what they're made to do, right? And so even though there's not enough moisture, because this area, it only rains every 15 to 20 years, and really never enough for enough constant moisture for the seeds to germinate. Okay? But the plants, they keep trying. All right? This is a female right here, making lots and lots of cones. What? It never rains enough. Why are you doing this? That's what things are programmed to do. Okay? People are like, oh, maybe these are the pollinators. No, if you know these little insects with the triangular backs, these are the piercing insects, and they're doing their thing to reproduce. And at the expense of the Welwitschia, they put their little eggs in here, and then their little larvae hatch and eat out the cone of Welwitschia, and somebody gets to reproduce. Okay? And then there's some ants. So maybe ants pollinate Welwitschia. They're here visiting the males, eating the pollen right there. Okay. For me, this picture alone was worth the macro lens I bought for this trip. Okay. There is climbing up there, eating the pollen. Okay. Ants make terrible pollinators because they don't fly. They're hymenopterans, but they don't fly, so they're not good pollinators. It turns out Welwitchias are flies are the pollinators of Welwitchias. They go to the males, they eat some pollen, and they're like little babies, right? They're really messy, and they go blah, 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 in there, okay? Here I'm trying to do it a little more carefully with a brush. I'm going in there collecting some pollen. And by the way, when you want to pollinate your plants, use natural bristles, like hair. Under emergencies, when I had longer hair, I would cut little pieces of my hair, and when I ran out of my good cheap brushes here. Okay, so I go in there and rub around, pick up the pollen on these. Anyways, pick up the pollen. Okay, And then what I do is I dangle the brush over the top of this and just sprinkle the pollen on so it lands on these little, what are called micropilar drops. This stuff is really sweet. If you ever see this on Welwitschia, I encourage you to take your finger, touch it, and taste it, and you'll taste the attractant to the flies for the female. Okay, nice sweet nectar. Anyway, I sprinkle the pollen on there. Okay, you know, it's a give and take. The plants give a little bit of the pollen, or the insects take a little bit of the pollen, but they're messy, so they eventually drop some of it off. Okay. And then that's not the same plant, but another one of my plants that we pollinated making seeds. There's a seed right in here. Okay, the seeds are winged like the seeds of maple trees and other things that have wings on them to blow the seeds to blow away. And when I take these seeds and I pull them off and plant them, I'll show you in a second the germinating plants. I tear off this outer wing, because that is more food for fungi and bacteria to get started and eat my seedling. Okay? So, yeah, and usually what I'll do is I'll take these and I'll dry them for a couple of weeks so that they dry on the outside. Again, less moisture there for fungi and bacteria, and then the seed kind of sets on the inside of there. Okay? So, we know we've been propagating Welwitschia for many years, and this last year I had a good amount of seed, so I decided to experiment again, you know? For the sake of science. And so here, I planted these in our succulent mix. All right, and you can see them germinating in there, the little cotyledons. And then these ones over here are in the sifted 1/4 inch red lava. Sorry, 1/8 inch red lava. 1/8 inch red lava, okay? So, you know, what's better for the plant? All right? Well, which one looks better to you? There's the soil on the left, and there's the red lava on the left. No, sorry. Red soil on the left and the red lava on the right. The lava, right? Seems to work a lot better. Yeah, it's more oxygen to the roots, to Welwitschia. Welwitschia suffer from lack of oxygen to the roots in pots, and so a really good draining mix is really important for them. Okay? It turns out when they get older, they can handle water, but and by the way, the ones that are missing there, we've sold off at some of our plant sales or the lectures that I do. So yeah, but guess which ones are harder to keep alive? The ones in the red lava and the pure red lava, these dry out. Welwitchias love moisture. Okay, so I saw that a few of you guys picked some up at the plant sales on last couple days. Do not let them dry out. Okay, if you have to, if you're not sure, if you have cooler temperatures, you're worried about them rotting out on you, a little bit of water, like Martha was talking about with the lithops, okay? Or I'm sorry, anyway, Jane, yeah. So, by the way, here, what's really cool about growing things from seed is you get oddballs sometimes, okay? There's the cotyledons of the Welwitschia, there's the 2 leaves that they make, and there's some extra leaves in the middle, okay? I've got about 3 plants like this that have extra parts on them. Okay? It's really cool. I'm waiting for one to grow like a normal plant and stretch out and have multiple leaves and internodes, but one day I can hope. Okay? Okay? Yeah, it's pretty cool, huh? I've got this last year from the batch, about 50 that I did, I got 1 variegated one. So I've got a variegated one, but I've got one that's got it's a double header. It's got 2 heads on it. Okay? Anyways, and I think, those 4. Okay? By the way, when you grow a lot of succulents, you get strange weeds. Okay? This is Aloe literalis that went into our gutter, so I call it Aloe gutteralis. Okay? Yeah, I think I'm going to call it gutter Alice from now on. Okay? Anyways, so there's a couple years ago, we got this grant to grow and propagate and distribute aloes because they're incredibly drought-tolerant plants. And so, anyways, to grow a bunch, and this is Kevin who's helping me out on the project. He got paid with a grant to do this. So he's a great help, and you'll see him in a little bit more later. And one of the things that I love about working at a university is that we get to teach the next generation and encourage students. And so he was one of this great group of these, what I call them, the like fantastic group of interns. So here he is pollinating Aloe. We're going to talk about aloe propagation now from seed. So he's pollinating the aloe, and see the tule cloth over here? He was the hummingbird for a little while here, and then he covered this up so that the hummingbirds wouldn't compete with him for pollination. Okay? To get pure Aloe. There's Aloe porphyrostachys, we're doing the same thing. Okay? I feel bad keeping the hummingbirds out, but there are plenty of other aloes around for them to feed on. Okay? Yeah, the tule cloth is a really good one for managing pollination. And also capturing seeds for certain things. Even for the aloes, it would help capture the seeds. So a couple years, I used to plant aloes seed with my limited knowledge of aloes and their needs. And I talked to a few of the aloe experts here, and it turns out that the aloes, their source of origins are tropical, and so almost nearly all species of aloes like good, nice, warm temperatures, 70, 80 degrees. Okay, and so here we were just trying out to see how these open pollinated seed that I collected, see how they would do. Okay, anyone here grow Aloe polyphylla from seed? Anyone? No? Okay, yeah, I wonder if Aloe polyphylla, which comes from cooler elevations, if it might be better with cooler temperatures for germinating, because I had limited success under my conditions, but I don't grow it because we can't grow it in the Central Valley very well. So here's the aloe seeds planted in pure red lava, because after talking to people, aloes tend to have problems with fungi attacking the seeds. So the seeds are like you see here, on the surface. We just plant them right on the surface. So fill up the pot with the 1/8 inch red lava, sprinkle the seed on top, water it in, bag it up, okay? And then once they germinate, once I see the little carpet of green, what I do is I start cracking the bag. What I like to do is I like to put slits in the bag with my nice sharp pruners and put a few slits in there, and then maybe even crack it open some more and basically wait to go from here, once they germinate, all that moisture in there, and just let it slowly over the course of a week or two get to this situation where there's no condensation in there. Now I can pull the pot of seedlings out into the air. Okay? So you never want to take them from here once they germinate and then open it up, rip them out, and put them out into the harsh, dry, harsh world. Okay? Low humidity world. You do it slowly so they can transition in.
Hey, Ernesto?
Yes, sir?
Did you ever do experiments about that?
Say it again?
Do you ever do experiments about that? Because I always do that, just take it straight out, and I've never had a problem in 20 years.
Here's the question that I challenge you, Brian. Did it slow the growth rate down of them or not? Right, they probably can take it, but if you do this slowly, will they go through a little less shock and have better growth? Okay, I don't know the answer to that, but when do you plant your seed?
I plant from March till October.
Okay, yeah, so you do it in the summertime as well, so the air should be pretty low humidity, right?
Well,
okay.
San Francisco, there's a lot of fog humidity.
There you go, that's it. You're where it's more constant, the humidity, the ambient humidity is much higher than mine. We get down to about 20%. And so I have to do this slowly. You're probably up at more like 40 to 50%, right? At least? That's probably what's going on there. Yeah. That would be my guess, okay? I'm not saying that's what happens, but... here's a bunch of them right here. We did a little experiment in the bags and out of the bags. And certain times of the year when we have more humidity in the greenhouse, I can get away with doing them straight on the red lava, okay? All right? But what I want to show you, well, I'll show you here in a little bit the details about the soil that we found out. But here's a bunch of different ones in here. Aloe melanacantha right here. This right here is Aloe bergensis. Ooh. Okay. Yeah. And I love this one when it's really young. Aloe microstigma. Actually, this is where Brian and I have no arguments that Aloe microstigma is one of the best aloes out there. Okay. So by the way, these, the question is are they ready to transplant? Okay? And the answer for most of these right in here is no. No, they're too young, especially the bergensis, okay? Too young. You have to let them get nice, big, and crowded. Let them get as big as possible so they both the above ground parts, the leaves and the stem above ground, and the roots are as sizable as possible, so that when you pull them apart, break them apart, you're actually doing You're not damaging too much of the tissue, okay? Minimal damage on the total mass of the plant.
Are you writing your labels with a felt pen?
Oh, no. Yeah, this is not just any pen. This is the Pilot SCAUF 45, okay? I'll show it to you later which one it is. We only write our labels with the Pilot SCAUF 45 or pencil, okay? Yeah. Do not write your labels with a Sharpie. Sharpies suck. Okay? Sharpies will wash away with regular water and they'll fade. This stuff is pretty close to permanent marker. Okay? Yeah. But pencil is even better, honestly. Yeah, pencil. You're a big fan of pencils, aren't you? All right, yeah. All right, you're my friend. Yeah. Okay, so I recently did a little experiment where I separated out aloes like when they were crowded, like in this pot here and in this pot, and I did them side by side at the exact same time. So I'm going to see what it does to growth rate of the plants. Okay, because my belief is that you should wait till they're like this and they do have less shock. But what I want to show you here is how we played around with the media that we actually grow them, that we actually planted the seed in. So this one over here is in straight succulent mix? Yeah, straight succulent mix. Bag soil, yeah, straight succulent mix. This is 1/8 inch red lava over succulent mix. Okay? This one right here is in pure, and also the red lava over succulent mix. These two are kind of in between. And then look at the one on the right, the pure red lava. So the thing was, I was convinced to use very inorganic material and the red lava for germinating the seed. But obviously this is better right here, right? Okay? But the issue when you have pure soil is that you can have a lot more fungal activity. And so this is what we found the happy medium. Succulent mix with a little bit of layer about an inch to a half an inch of the sifted red lava on the surface. So the seed are sitting on top of that inorganic layer. Once they germinate, they go through and they get into the soil where they do better.
Red lava is better than the black lava?
They're very similar in composition. So, you know, by the way, you can't, not everybody here can get red lava, right? So you find a material that's suitable, some kind of inorganic rock that's kind of semi-porous, and use that instead. Okay? Locally source what you use.
What about pumice?
Pumice, you know, we used to use pumice, and it worked really great. But a few years ago, we lost our source of pumice. And you know, I like to, when this gets recycled into the soil, it doesn't make my soil with all of that white speckly stuff. But really quick here, it turns out that red lava is about 20% less porous than pumice is. Pumice holds more moisture. So when you have things in pure lava, you need to water a little bit more because it's actually a lot drier. Okay? So they both work. And when they're in the bag, it doesn't really matter. But we use the red lava now because we have a better source of it.
Okay, it's pretty easy to get pumice in like Washington and Oregon.
Yeah, and in Southern California, in volcanic regions. But in Central California, it's a little bit harder for us to get it now. So yeah, there's places, probably back east, you guys don't get pumice, do you? No, yeah, you have to find something else. Okay? So yeah, that was just to show you the differences and see what we found out is that this was really good for germination, but not so good for growth rate of the plant. Okay, and so that's just experimenting to find out what sort of maximizes all your variables for growing your plants. Okay. All right. There's just a bunch of the seedlings that we planted, a bunch of different species. I love doing this, growing a bunch of the different related plants together to see the differences even from the baby stages, okay? That's kind of cool, right?
Yes.
All right, it's fun. So try that out, okay? Yeah. By the way, Tim Harvey, who's in Thousand Oaks, he can get away with germinating and growing some of his aloes when they're really young in pure perlite. Okay? So, you know, that's pretty amazing. And then here, this is in a chamber that he has a cold frame that you can control the humidity in there by opening and closing this door, and it's covered and shaded to basically make a big humidity chamber. Okay? So, you know, the Ziploc baggies, I do try to recycle them. Okay? So I don't slit, I minimize the slitting technique. I usually open the bags up, open the top so I can reuse them, so students can take away used soil and stuff like that. Okay? Okay, a little bit more about pollination and seed and stuff. This is not, this is rough pollination right here, okay? Tim Devine up at Chico State, he's taking his male plant and he's rubbing it up against the female, okay? I do not recommend this technique at home, okay? Use a brush instead, okay? Euphorbia obesa are typically male or female plants, but every once in a while you get some that are both. Male flowers, actually male inflorescences technically, and then females in here. Okay? Yeah. So this one, yeah, selfing. I can just pollinate, it can pollinate itself. By the way, going back to Dorstenia gigas, one thing that I found out is that you typically, it doesn't seem to work that you can self-pollinate Dorstenia gigas. You need genetically different individuals to pollinate. Okay. Anyways, here they made the fruits. I put them in a dry aquarium with a cover on top so the seeds can bounce around, and there's some seeds right there. Okay. This is very like, you know, some of them can land in here, and you know, they either land in here or they land down here. And just by virtue of the amount of area that you have open, most of them are going to land down in here to collect. But this is what Isabel over in near Fullerton, what she does is she made these little cloth from like shade cloth, and she made these little I don't know, funnels, if you will, where the seeds bounce around there, and most of them get caught in this little folded area in here, and then she takes them and dumps them out and into coffee filters to dry them out. Kind of cool, right? Okay. A number of years ago, with tule cloth, I pollinated. I had 2 clones of Euphorbia abdelkuri. This was back in 08. And look, 9 years later, 6-inch tall seed-grown abdelkuri. That's slow. This plant's slow. My adult plants, I'll show you later, I tried watering them a little bit more to get them to grow faster. Big mistake. I have to basically make cuttings of mature plants to replace the root system that I rotted out. Yeah. Another Euphorbia jatropha. Okay, there's my. Oh, you guys know these little wedding favor baggies? I love these. These are great. Yeah. Okay. It's like. Da, 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 da. Okay. Anyway. All right. So I want to show you this because a friend of mine did this with, someone was talking about the, you know, Astrophytum asterias, okay? I want to show you this, a little technique that he showed me for planting the seeds of this that would probably transfer to other cacti and probably a lot of other succulents as well. So clean seed, pull them out of the fruit, nice and clean seed, okay? And back here you can see a little bit of the diversity. We'll see a little about the more, oh, there you go. Look at this. He's trying to make his own super kabuto. Right here, style 1, and look at this one. This is so cool. The ones that have less spotting, and then the winners in here. Okay, yeah, and this is the beauty of growing stuff from seed. Look right here. Are you my brother? Okay, or sister? Are you my sister? Okay. So here's what my friend Barry Rice does for growing his astrophytums and other cactus seeds. This is not pure red lava. This is actually the succulent mix. Okay. He sprinkles the seeds on the top of the succulent mix, covers it with sand, and then waters it with the hydrogen peroxide. This is 3% hydrogen peroxide. Okay, it's not really super concentrated stuff. So he waters it in and just soaks it, and basically what the hydrogen peroxide does, it probably destroys a lot of algal spores, fungal spores, other spores, you know, yeah, fungi, algae, and probably mosses as well. And then basically sterilizes the soil, and then he puts them in baggies. Okay? So there are his baggies back in here. Okay? He does them in the little rose pots, the smaller pots, because he doesn't have to do big batches like I do. And then he does the same thing with the baggy technique and opening them up, etc. Okay? And so I want to show you this here. This is 23 days after germination using the hydrogen peroxide. By the way, you see how his soil sunk down from the original height? The hydrogen peroxide ate up the organic matter and broke it down, and it sunk down in there, because that's what hydrogen peroxide does. Okay? And there's 75 days later. Okay? And then 6 months later. Okay? And so he says that he debags them about 4 to 6 months later when the following will occur, but I just want to show you this. He says now that he's using the hydrogen peroxide, when he waters them in, a lot less algal problems. Okay? So it does sort of...
One hydrogen peroxide watering or more than once?
Well, just once. Just the initial one. But then he's got them in the baggies, so he doesn't need any more water after that. Okay? So, yeah, maybe you could do that with your cycad seed. All right? Anyways, this is what he says. He basically keeps them bagged until he sees the algae starts growing, thickly and over the plants. They're about 8 to 10 millimeters in diameter, or when he's out of room. Okay? Yeah. So when basically he needs room for his next batch of seedlings. But yeah, that's a pretty good amount of time in the baggies without any extra water. Okay? I don't know, maybe 4 months later he goes in and dribbles a little bit more water, but pretty much not. So there it is again all together. Okay? And what Barry was telling me, he's talked to some people. There are some people that use fungicides on a very regular basis, okay, or maybe better sterilization techniques like our soil at UC Davis, and they keep them bagged for up to a year. But I myself, I don't like to use this stuff on a regular basis. Okay? In fact, I never plant seed with fungicides. Maybe here and there I should, but I don't like to use a lot of chemicals. And so that hydrogen peroxide is a good happy medium.
Ernesto?
Yes?
Mauro Peixoto down in Brazil uses chamomile tea.
Oh.
When he plants.
Okay, so it has some kind of antifungal properties to it, probably? Yeah. Yeah, probably there's a lot other plant materials that you could use to do that same sort of thing, yeah. Yeah, apparently creosote, so those of you that are in the Sonoran Desert, creosote is supposed to have some antifungal capabilities, so maybe you can make a tea from that. But don't drink it out before you do some research on that, okay? All right. By the way, letting the seedlings get crowded, okay? Ooh, I better speed it up here. Letting the seedlings get crowded is pretty important, okay? These ones got a little bit too crowded. All right, let's get on to some tissue culture here to finish up in the last 5 minutes. Okay, so we were trying to do some tissue culture with aloes, and we were using aloe flower stalks. What basically we found out is that let Tim Harvey do the tissue culture. He's better experienced than we are. Okay, here's his lab right here. We tried it, we just didn't do it long enough within a grant cycle to get really good at it. Okay, even Tim, when he first started out, had to play around with the techniques to get better and better at it. Okay. So those of you that buy his plants, he puts a lot of energy into making those plants happen. So, yeah. A little public service announcement, okay? Alomite, someone mentioned it, brought it up before. Watch out for this on your aloes, all right? If the best method of getting rid of it is removal, physical removal. Okay, and I just brought this in because sometimes this is like material for tissue culture. If you kill off the mites, then you can use this for tissue culturing because it's proliferation tissue. Anyways, for killing off the alomite, if you want to use a pesticide, miticide called Contos works really well. Okay, there's the mite. There it is, the LA arboretum attacking many, many aloes. Okay, watch out for it. It also jumps on your relatives, like asterias and haworthias. Okay. What did my dinner get in there? Oh, okay. Okay, I'm going to jump ahead and to finish up in the last 5 minutes here real quick. So I want to show you this vegetative propagation technique with aloes and quartering. Some people carve them out. I like to quarter them. Okay. March 2001, a couple months later, we tried using cytokinin to get it to proliferate more. Very bad idea. Aloe kalanzai is very slow growing. When you add extra hormone, it's a fail. Okay. See this one over here? This one here that has 4 heads. Each one survived. This one, the ones that I treated with cytokine and didn't grow, and I lost the other two here. I want to show you, and by the way, when you're quartering them, use a nice sharp knife, stainless steel, sterilized, not an unsterilized serrated knife. Rot. Okay? Yeah. There's the plant when it grew up. I just took go back recently. And look at this. I split this one again, so I'm making a shrubby Aloe Palanzii. Okay? Yeah. So the quartering technique. You can quarter them because this meristem is made up of many cells, and if you split that and then keep it apart, then each one will grow individually. Okay? I want to get to tissue culture grafting real quick. So we were doing this with a couple of aloes like Carisbergensis and Burrii. I used the late greenhouse labels to split them apart and keep them apart from each other. Okay? This one just happened by an accident. Rainfall damaged the growing meristem, knocked it off. It didn't rot out on me, and now this guy is making babies. So sometime nature will make more plants for you. Look at this one. You guys know this one, right? Ooh. Okay, I finally decided to try this. Stainless steel knife. Okay, and each one of these should grow and then proliferate and make more babies for us. So, try this or don't try this at home. Okay? All right?
Why did you do that rather than drilling?
Because to me this is a little bit more controlled. You do less damage. It's like anyways, it's like a surgery versus I don't know, a puncture. Yeah.
Ernesto,
Yes.
on the Palanzies, you split them, but you didn't separate them like you did the originator. It's that what do you use to hold them apart?
The greenhouse labels. I just wedged greenhouse labels in between them. Yeah. Because the aloes, I wanted to keep them on the plant, and so I just split the meristem but left it attached. Yeah. But Rob Roy down in Fullerton told me that you can go through the whole plant here and it'll be fine. So I took his word on it. Cacti, same sort of thing. Split the meristem. Now I have 2 branches instead of 1. Okay. And with the cacti, you have to cut the tops of them off and then splay them apart. If you leave the tips on them, they'll grow back into themselves in the middle. So you have to chop the top off and then get it to branch. Let's get to grafting real quick. Hormones. By the way, if you ever use hormones on succulents, use really low concentration. Succulents are very sensitive to hormones. They just need a little bit to direct them, okay? See, well, I'm just going to jump ahead. I want to get to tissue culturing real quick. Dorstenia gigas, propagated via aeroponic propagation. Look at that, beautiful root system on there. Okay? Yeah, I'm going to jump ahead. There's my Aloe abdelkuri, Euphorbia abdelkuri cutting. There's the plant that I had to reroot. Bad, Ernest. Okay? See this right here, when you make cuttings of cacti, you don't want to plant them right away with this. Keep them upright so that the hormones work really well and travel down to the bottom. Wait till the wound heals like this, where it's really leathery, and then there's roots there. Now it's time to plant it in some lava rock. Okay? I don't plant them into soil right away. Okay, let's go ahead and jump ahead to that lovely plant that people want, to grafting, okay? Ooh. Yeah, Stechum hintonii. Okay, I'm just going to do the grafting and then I'll stop. So we grafted these guys right here to speed them up, and look how quickly, from germination to grafting 2 months later to the plant flowering within 1 year. Okay, that's the beauty of grafting. So just let me show you the techniques real quick. When you do this, when you graft cacti, you can use rough instruments like a nice sharp pruner to cut the top off of this one, cut the bottom off of this guy right here, and then set it on and then clean it up with a really nice razor blade, really nice sharp razor blade that's sterile. You can reuse it or sterilize it or get a new one, and then you set them on top, line up the vascular tissue, stick them together with like a rubber band. Okay, and there it is. You have to play around with the rubber band technique really well. Okay, and there it is right there, you know, growing. We put them, when we do this grafts, you put them somewhere humid for a few days, for about a week, so that the wound heals really slowly and the two can connect up into each other and you get connection being made. Okay? If you ever graft a Stechum hintonii, keep it stuck on there because it tends to grow cells underneath there and push itself off. Okay? So we use and this is another one right here, Teflon tape. Okay? So you put them somewhere humid for just a few days till they connect, and then you pull them out of the humidity chamber so they don't rot on you. And there they are growing on their own. Okay? And it's a great way of rescuing plants whose root systems rot out. Okay? We had this happen with the Astecium hintonii, actually Astecium ritteri. I got one of those ones that was from UC Berkeley that they took in a confiscation, and I almost lost it on its own root system, and I grafted the last little bit that I had, and it's a beautiful specimen now. Okay? Because I saved it, so this one was rotting out on me, this crested Epithanantha micromeris. Frankenstein plant right there. Okay? Thank you very much.